Welcome back to the third in our series of carbohydrate lectures. We're gradually filling up our overview picture of carbohydrate metabolism. We've covered this bit up here, the structure and properties of carbohydrates and how they're transported into cells. And in the last lecture, we looked in detail at the regulation of glycolysis. But in this lecture, we're going to look more specifically at the TCA cycle and how that is coupled with oxidative phosphorylation to generate a whole stack of ATP. In addition, we'll look at how the glycolytically produced NADH is linked with oxidative phosphorylation by these electron shuttles. So let's start with the TCA cycle. And to function, this pathway requires oxygen. As such, it is also referred to as an aerobic pathway, underpinning aerobic metabolism. This type of metabolism is far more efficient than anaerobic metabolism. And therefore, when dealing with issues of efficiency in production animals, this pathway is a key player. In addition to that, some toxins are known to inhibit this pathway, effectively inhibiting aerobic metabolism. And chronically, this can be quite lethal. So on its own, this may seem like a fair bit of detail, but this pathway is just so crucial to so many aspects of the normal day-to-day function of animals. The TCA cycle is also called the citric acid cycle, or the Krebs cycle, after the fellow that discovered it. And this pathway is quite crucial. It's really important for oxidizing carbohydrates, lipids, and proteins, but also it's crucial because it's a major point of intersection for the substrates of a whole stack of different pathways, including gluconeogenesis, transamination, and deamination of amino acids, and also lipogenesis. And you find it expressed exclusively inside the mitochondria, which is one of the organelles inside of your cells. Now, there's a fascinating story about mitochondria. Did you know that they've got their own DNA? So the story goes that way back in the day, the mitochondria were actually a separate cell. Now I'm talking way back during the Precambrian period when there wasn't much oxygen kicking around in the atmosphere. So cells at that time were all anaerobes, existing in the oceans, creating energy from glycolysis. Finally, the early photosynthesizers, so these were the early plant cells, started oxygenating and creating the modern atmosphere. And then another cell worked out that it could make use of this oxygen to further metabolize the fermented byproducts of the anaerobes. And this cell was the ancestor of the modern mitochondria. Now, this symbiosis became so convenient that one cell hopped inside the other, and hey presto, we've got the modern aerobic cell. Now, I do concede that's all a bit theoretical and maybe a bit hypothetical, but it is one of the plausible explanations for how we've arrived at these two components of a cell that have their own mitochondria. So let's have a look at the TCA cycle in detail. And if you're excited about such things, there is actually plenty of detail there. Here's a diagrammatic representation of the pathway with the enzymes named and all of the substrates, which are the intermediates. But I can completely assure you, you don't need to remember all of this. It will not help. Let me show you simplistically what I do want you to learn. Here's the level of detail that actually matters. This pathway metabolizes carbon backbones sourced from carbohydrate, protein, and lipid. Through their breakdown, they are converted to acetyl-CoA, which is a 2-carbon molecule, and it enters the TCA cycle here, condensing with oxaloacetate, which is a 4-carbon molecule, giving you citrate, which of course, if you add 4 carbons and 2 carbons, you're up to 6 carbons. And that's catalyzed by citrate synthase. So there you go, you can learn this enzymatic step and the associated carbons. Now, citrate, through a series of other enzymatic steps, is gradually broken down It releases two CO2 molecules, so in effect the two carbons that went in here are cleaved off at a couple of different points around the cycle. And it also generates a whole stack of NADH, 3 of them in fact, FADH2 and GTP. And after you've done one complete lap of the TCA cycle, you're back to your starting molecule, oxaloacetate. So in some ways, you can think of oxaloacetate as being a bit like a carrier molecule or a taxi. It picks up a passenger, acetyl-CoA, and gradually catabolizes it on the way around this pathway. And then the taxi gets back to the starting point as a 4-carbon oxaloacetate molecule. So there you go. That's the level of detail that you need to memorize about the TCA cycle. Let's next have a look at what we do with these mitochondrially produced reducing equivalents. Through oxidative phosphorylation, which also occurs in the mitochondria, these are converted into ATP. So let's have a look at the detail of this. First of all, the pathway itself, it occurs inside the mitochondria, and it couples respiratory oxidation to ATP generation, making use of the respiratory chain, or the electron transport chain as it's otherwise known. Now this is simply a series of catalysts, and I could go into a heap of detail showing you what these look like as molecules, but there's really not a lot of point. You just need to know that they are there and that they collect and transport the reducing equivalents, so those are NADH and FADH2, directing them to their final reaction with oxygen to form water. And of course, the production of a whole stack of ATP. So here's the level of detail that actually matters. First of all, I've represented the respiratory chain here. And yes, I've shown you no detail at all. At the moment, it's just a concept. Over here, we've got the TCA cycle producing NADH and FADH2. And the electrons from these are transferred onto the respiratory chain, generating ATP from ADP, ultimately using oxygen as a final electron acceptor, producing water. There are other sources of NADH and FADH2. For example, beta oxidation of fats, which you'll hear about in the coming lectures, is a big source. And I've already mentioned how glycolysis itself produces a molecule of NADH. But the other thing to realise is that per molecule of NADH, you yield 2.5 ATP via the respiratory chain. Whereas if you are generating FADH2, you only yield 1.5 ATP via the respiratory chain. Now these may seem like funny numbers, 2.5 and 1.5. And the reason why I've represented them as these estimates is that the respiratory chain itself has a certain level of inefficiency or uncoupling. And metabolic studies have therefore defined the ATP yield as 2.5 and 1.5 units from these reducing equivalents. The other thing that sometimes gets overlooked here are the outputs or the end products of this reaction. The key ones are carbon dioxide and water. Now, to excrete these waste products, well, you can sweat or piss this stuff out, and for CO2, well, you breathe it out. So stop to think about that for a minute. Where did the fuel come from for these pathways? So far, I've shown you how carbohydrates can contribute to these pathways, providing glucose. And of course, the glucose itself is ultimately sourced from your diet. Lots of things have got carbohydrates in them: bread, pasta, cereals, potatoes. So when you think about it, you're eating carbohydrates, or you're eating the spud. It gets digested and absorbed, releasing glucose into your bloodstream. This gets broken down via glycolysis initially, and then via the TCA cycle, producing carbon dioxide and water, which you piss and sweat out in the case of water, and breathe out in the case of CO2. Isn't that a weird concept? You eat the spud,
and then you effectively breathe it out via CO2. Amazing stuff. That's what first got me interested in biochemistry.
So here's the really detailed depiction of the TCA cycle and the respiratory chain. And like I said before, you don't need to know it at this level of detail. This is just for those that are excited about biochemistry and want to look at it in more complexity. Now, based upon the reducing equivalents produced, we can actually calculate how much ATP is generated from one molecule of acetyl-CoA. So first of all, one lap of the TCA cycle produces 3 NADH molecules, 1 FADH, and you'll remember that funny molecule named GTP. That can also yield 1 ATP directly via substrate level phosphorylation. These other two, so FADH2 and NADH, are of course linked to the respiratory chain, producing 2.5 ATPs and 1.5 ATPs. So when you add all of that up, you end up with a total of 10 ATP produced per molecule of acetyl-CoA. So with this extra energy output, this suddenly starts to give you a clue as to the efficiency of aerobic energy production.
Many of you will recall how glycolysis produced a molecule of NADH. In fact, it produces 2 per molecule of glucose. I'm now going to show you how this is shuttled into the mitochondrion so that it can be coupled with oxidative phosphorylation for the generation of ATP. And this is done with the use of substrate shuttles. So first of all, let's recap. You'll recall how in anaerobic conditions, NADH is converted back to NAD+ via the production of lactic acid. But the question is, how do we recycle this NADH in aerobic conditions when oxygen is present? Well, simplistically, this is shuttled into the mitochondrion via a substrate shuttle so that it can be coupled with oxidative phosphorylation. But let's have a look at the detail of how these substrate shuttles work. Now, to understand these substrate shuttles properly, the first thing to realise is that the inner mitochondrial membrane is relatively impermeable. Small molecules like oxygen and water and CO2, they can pass freely across that inner mitochondrial membrane, as can acetic and acetoacetic acid and 3-hydroxybutyric acid. And these are all useful aerobic fuels, which I'll tell you more about later. But down here, you can see a list of notable substances that do require specific transporters to pass through the mitochondrial membrane. And this includes things like pyruvate, also citrate and isocitrate, and a number of other intermediaries of the TCA cycle, and also ATP and ADP. But in the context of this story, the key substrate that isn't permeable across the mitochondrial membrane is NADH. And so, of course, that's a problem for the glycolytically produced NADH. We can't link it to mitochondrial oxidative phosphorylation unless we can get it into the mitochondrion somehow. And this is where the substrate shuttles come into play. There's 2 of them that I'm going to tell you about, the glycerophosphate shuttle and also the malate aspartate shuttle. Let's have a look at these 2 in more detail. So let's have a look at the first shuttle, the glycerophosphate shuttle. This is a bit of a stylized representation. Here we've got the inner mitochondrial membrane and the outer mitochondrial membrane. And then outside of that, we've got the cytosol of the cell. And down here is the glycolytically produced NADH. So the first step is to transfer the electrons from NADH onto dihydroxyacetone phosphate, forming glycerol 3-phosphate. And this is catalysed by this dehydrogenase here, glycerol 3-phosphate dehydrogenase. Glycerol 3-phosphate can then permeate into the mitochondrion via a transporter, and then this same enzyme, glycerol 3-phosphate dehydrogenase, catalyses the reverse reaction transferring the electrons onto FAD, producing FADH2, and recycling dihydroxyacetone phosphate so that it can pass back out of the mitochondrion, ready to do another lap of this cycle. And this keeps going around and around, transferring electrons from the glycolytically produced NADH into the mitochondria so that via FADH2 they can be coupled with the respiratory chain, producing 1.5 ATP per molecule of FADH2. The next shuttle I want to describe is the malate aspartate shuttle. And in this case, it's a little bit more efficient than the glycerophosphate shuttle. And that's because the cytosolically produced NADH from glycolysis is coupled with the production of NADH on the inner side of the mitochondrion. And of course, this leads to the generation of 2.5 ATP. You'll remember that the glycerophosphate shuttle was coupled with the production of FADH2 inside the mitochondrion, which only produced 1.5 ATP. So there's a bit of an efficiency difference here. Now this pathway may seem a little complex, but let me break it down for you. First of all, the NADH transfers its electron onto oxaloacetate, forming malate, and this is catalyzed by a dehydrogenase enzyme. Now malate can transport into the mitochondrion via an exchange transporter, alpha-ketoglutarate exchange transporter here. The reverse reaction then applies on the inside of the mitochondrion, again with malate dehydrogenase in action, producing NADH and recycling that molecule of oxaloacetate. Now to get this back out of the mitochondrion, we transaminate it. In this case, we rob glutamate of an amine group, dropping it onto oxaloacetate, forming aspartate. And the carbon backbone of glutamate gives us alpha-ketoglutarate, which then couples via the exchange transporter to swap for a molecule of malate going in. So we've got alpha-ketoglutarate on the outside of the mitochondrion. And aspartate is exchanged for a molecule of glutamate coming in via this exchange transporter. So we've got aspartate on the other side of the mitochondrion now. And we can then simply reverse the transamination reaction, dropping the amine group back onto alpha-ketoglutarate to give us glutamate, and aspartate, if you rob it of its amine group, becomes oxaloacetate, ready to do another lap. So in effect, if you stand back and look at this, we've got two big intersecting wheels. The top wheel going around here, and then a bottom wheel going around here, intersecting in the middle. And of course, the crucial outcome here is that the electrons are shuttled into the mitochondrion for ATP production. Okay, so with this in mind, we can now look at the total amount of ATP produced per molecule of glucose in an aerobic scenario. We've already worked out that glycolysis itself directly produces 2 ATP. In addition to that, glycolysis produces 2 molecules of NADH per molecule of glucose. Now, if we assume that we use the malate aspartate shuttle, this means that the 2 NADH will be coupled with the respiratory chain once they've been shuttled in, giving us another 2 NADH, giving us a total of 5 ATP. We've also looked at the activity of pyruvate dehydrogenase. It also produces a couple of NADH molecules, which are coupled with the respiratory chain to give us ATP. And then lastly, the TCA cycle itself produces another 10 ATP from that combination of NADH, FADH2 and GTP, which it produces. So therefore, we've got 2 glycolytically produced ATP, 5 ATP from the substrate shuttle, 5 ATP from pyruvate dehydrogenase activity, and another 20 produced from the TCA cycle, giving us in total 32 ATP per molecule of glucose. Of course, if you're using the less efficient glycerophosphate shuttle, then the total amount of ATP produced will be 30 instead of 32. Now this would make quite a good question, don't you think? If I was to ask you how many ATP do you generate per molecule of glucose in an aerobic scenario, the answer would be 32. But if I was to say how many did you produce in a purely anaerobic scenario, then the answer would only be 2, because you wouldn't get the contribution from the aerobic steps of this pathway. You can start to see the massive efficiency difference between anaerobic versus aerobic metabolism. For anaerobic metabolism, it's 2 ATP per molecule of glucose, and for aerobic metabolism, it's 30 to 32 ATP per molecule of glucose. This has quite important implications for exercise. Have you ever heard of training regimes structured around an anaerobic workload versus an aerobic workload? Well, in effect, they're referring to this. If you want to run a marathon, you better make sure that you maximise your aerobic output to make your fuel last as long as possible. But if all you're doing is sprinting 100 metres, then you don't need to worry about the efficiency of ATP production, and you can burn through that fuel far quicker using anaerobic metabolism. And the other key advantage of anaerobic metabolism is that glycolysis, which is the pathway that underpins it, can be accelerated massively. In fact, it can yield ATP up to 10 times faster than the TCA cycle when it's been accelerated maximally. And so therefore, anaerobic glycolysis is absolutely crucial when you're either sprinting or if you're in a fight or flight response. This is the pathway that's the key factor responsible for why you've probably survived and reproduced across the eons. We've had a good glycolytic pathway to enable us to escape from lions and tigers. So in summary, we've looked at the TCA cycle, which is the final pathway for oxidising carbohydrates, lipids and proteins, and it produces NADH and FADH2. We've also looked at oxidative phosphorylation, which generates ATP from NADH and FADH2, and this pathway uses oxygen as a final electron acceptor. And that's why this combination of the TCA cycle and oxidative phosphorylation are considered to be an aerobic pathway. And we've looked at how substrate shuttles can transport the NADH produced from glycolysis into the mitochondrion so that we can generate ATP from it as well. And therefore, we can produce a total of 30 to 32 ATP from one molecule of glucose catabolized aerobically. And that's a massive efficiency difference compared to anaerobic metabolism, which can only produce 2 ATP per molecule of glucose. And to help with your study, and just to make sure you've captured the right information from this lecture, I've got a series of 4 questions which you can help to focus your learning objectives.
The next step in our carbohydrate series is the pentose phosphate pathway. So returning back to our big picture of carbohydrate metabolism is the pentose phosphate pathway, which is this bit here off to the side of glycolysis. And one of the crucial roles of this pathway is to produce reducing power in the form of NADPH for fatty acid biosynthesis. And the other crucial role of this pathway is to produce ribose residues for nucleotide and nucleic acid synthesis. So that's effectively acting as the backbones for RNA and DNA production. Now this pathway occurs in the cytosol of cells, and you can see down here there's a summary of that pathway, but I'll go into that in a bit more detail in a moment. The other key point to recall is that this pathway produces NADPH for reducing power so that we can generate fatty acids and steroids. So in this case, NADP plus is the electron acceptor, not NAD plus. So it's a crucial thing to note. Now NADPH has a number of crucial roles within your body. I've already mentioned that it's crucial for fatty acid synthesis and fatty acid elongation, but there's also a number of other molecules that it's crucial for the generation of, including cholesterol, neurotransmitters, nucleotide, and superoxides. It's also got a crucial detoxification role. So there are antioxidant systems within your cells, and NADPH enables the regeneration of these systems, in particular targeting the reduction of oxidized glutathione, regenerating it, and also cytochrome P450 monooxygenase. Now, the pathway itself consists of two key phases. There's what we call an oxidative phase and a non-oxidative phase. The oxidative phase is a series of non-reversible dehydrogenation and decarboxylation reactions, and these produce the NADPH and ribulose 5-phosphate. The non-oxidative phase is a series of reversible reactions, and they produce ribulose 5-phosphate, which can be converted back to glucose 6-phosphate. So let's have a look at this pathway in a bit of detail. Now, this picture looks a bit scary at first glance, but let me break it down to you and show you the simplicity. First of all, at the top here, we've got the oxidative phase of this pathway, driven by these dehydrogenation enzymes. And then in the bottom half of this picture, so below this line running through here, we've got the non-oxidative phase of this pathway. Now, it's in the oxidative phase where all of the NADPH is produced at the top. And in this case, we take glucose 6-phosphate from out of glycolysis and convert it into ribulose 5-phosphate. And you'll note that this step has been repeated 3 times across the top here. Now, the reason why is that once we get down to ribulose 5-phosphate, the non-oxidative phase of these pathways is basically a series of rearrangement reactions, shuffling the carbons around until we get the molecule back into its original glucose 6-phosphate form. But the key thing to note is that we started with 3 molecules of glucose 6-phosphate, and by the time we've finished, we end up with 2.5 molecules of glucose 6-phosphate. And the crucial output from the non-oxidative phase of this pathway is the production of ribose 5-phosphate, which underpins the synthesis of nucleotides like RNA and DNA. So, in summary, we've got an oxidative phase at the top, potentiated by these dehydrogenase enzymes producing the NADPH, which is the reducing power that cells require, particularly for fatty acid biosynthesis. And then we've got the non-oxidative phase at the bottom, which basically takes the end products, ribulose 5-phosphate, and rearranges them back to their starting form of glucose 6-phosphate. And the regulation of this pathway is crudely simple. It's via NADPH, which acts as an allosteric inhibitor of glucose 6-phosphate dehydrogenase. So in effect, it's this enzyme up here. So that's basically end product inhibition, slowing it down when the production of NADPH is too high. And I just thought I'd briefly remind you where we got that substrate from for the pentose phosphate pathway. It's glucose 6-phosphate, which is this substrate here, the first intermediary of the glycolytic pathway. Now, the pentose phosphate pathway is expressed to higher levels in particular tissues, especially those that are involved in the synthesis of fatty acids, steroids, amino acids, or reduced glutathione. All of these processes require a whole stack of NADPH for their reductive biosynthesis. So the tissues that have a big role in this biosynthesis process include the liver, the adipose tissue, adrenal cortex, the thyroid, testes, erythrocytes, and of course the lactating mammary gland. But not all tissues require heaps of reducing power, and therefore not all tissues need lots of NADPH. And yet, most tissues still need ribose because it's acting as the backbone for DNA and RNA. So in those tissues that don't need a lot of reducing power, The oxidative phase of the pentose phosphate pathway through those dehydrogenation reactions is only weakly expressed. So for these tissues, rather than bothering to express those dehydrogenation enzymes, all they do is reverse the bottom half of the pentose phosphate pathway, that is the non-oxidative phase of that pathway. If you reverse it, you can produce all of the ribose 5-phosphate that you require. So in summary, I've shown you that the pentose phosphate pathway provides reducing power and ribose backbones. It's got an oxidative phase, which is irreversible and underpinned by a couple of dehydrogenation reactions. And it's also got a non-oxidative phase, which is entirely reversible. And for those tissues that don't need NADPH, it acts as an alternative avenue for producing ribose so that you can make DNA and RNA.
And just to recap, here are a series of questions to underpin your learning about the pentose phosphate pathway.